Thanks, Matt. This is my second year presenting here, so if you came last year, the first part of this talk is going to seem like a repeat because we're using actually the same group of animals, the same data set, but we have some additional results at the end. So if you were here last year, you just have to go through a little bit of repeat here. So for the background, we're looking at reproductive efficiency, and as many of you know, reproduction is very important in the cattle industry. Reproductive efficiency is determined by lifetime production of the cow, and it's an important element in cow-calf, an important element of the cow-calf component in the cattle industry. Because it would be great if we, before you breed your heifers, if you knew which ones were going to get pregnant and those that, do not have a chance of being bred, and put them in a different pathway, for example, going to the feedlot instead of attempting to breed them and putting a lot of effort into them for reproduction. So our objective was to identify regions of the genome that are associated with reproductive efficiency in beef cattle. Now there are some challenges when you look at reproduction. First, it's a lowly heritable trait, and it's a very complex trait also, which means it's influenced by multiple genes, and therefore this creates difficulty when identifying genomic regions. Now one way to overcome these challenges is to look at a large number of animals. And I'm not talking hundreds of animals, I'm talking thousands of animals. And when we started this project, obviously genotyping animals, it can be expensive. When you're looking at the 770 chip, cost $185. And for this project, we utilized over 6,000 animals. So that's the largest sum of money if you're going to genotype each of those animals individually. And so one of the scientists here, John Keel, had been reading some research, and they were actually using DNA pooling and then genotyping the pools as a way to decrease cost when utilizing the SNP chips. And so that's what we did, where we actually pooled based on the extreme phenotype which reproduction gives us, because you're either pregnant or you're not pregnant. So this is just an example of what we did. This is for the MARC population, where we had low reproductive animals and high reproductive animals, so those were our two extremes. And we selected 1,000 low and 1,000 high animals and pooled 100 of those into an individual pool. So we had 10 pools for the low and 10 pools for the high, and then genotype these on the 770 chip. So just to give you a brief introduction to the populations that we evaluated, the first one was the MARC population, which was, I showed in the previous slide. And we have a very genetically diverse population, which many of you know. We have both British and continental breeds. And again, we took 1,000 low and 1,000 highly reproductive animals to evaluate. We then had access to a population in Central Florida, and the high points of this population was they have Brahmin influence. They're 3/8 Brahmin in their breeds with Brangus, Simbra, and Brayford. And then they also keep their animals around for 2 breedings. Like a lot of commercial ranches, once you have an open animal after 1 breeding, she's gotten rid of. They actually keep them around for a second breeding. So we had an excellent extremes here. We had open animals, and we had pregnant. And then of course we have our in-betweens where they were open pregnant or pregnant open. And then we also worked with the ranch out in western Nebraska, where we had records from 2007 born animals. And again, we had 2 extremes. We had animals that were open after 1 breeding, and then we had animals that had 3 consecutive pregnancies. And then finally, we worked with Milt Thomas, who at the time we did this research was at New Mexico State University, he's now at Colorado State University, and he had a couple smaller populations with some Brahmin influence. Now, if you recall my talk from last year, I presented a lot of these plots here. This is actually a Manhattan plot. And what we did for the plot I'm showing here is we actually combined the data from all 4 of those populations and got the following plot. So we have the chromosomes along the bottom here, going from 1 to 29, and then we have the sex chromosomes. Now, if you remember from last year, I talked about the Y chromosome. Now, we've taken that out just because it was so high, and we actually wanted to look at other regions across the genome. So what we have here is each dot represents a SNP on that chromosome. when that SNP is associated with our trait, reproductive efficiency, and is above this line, it's significantly associated with our trait of interest. And as you can see, we get a big peak on chromosome 5, 13, 15, 18, and 29. So what we wanted to do and what I'll present today is actually going into chromosome 5. Obviously, this had the highest significance and the largest region associated with reproductive efficiency. And there's actually been some papers that have come out, in fact, one this year, where they have also found regions on chromosome 5 associated with reproductive traits. So, we have this data. We want to look further at chromosome 5, and there's a number of things you can do to evaluate what could be causing this association with reproductive efficiency. One of those is to do some fine mapping. We've already done whole genome association, where we look at SNPs along the genome. And for example, as we showed for chromosome 5, we're able to identify a region associated with our trait. So to further evaluate that, one thing you can do is to look further in that region of significance and do some fine mapping. And by fine mapping, we basically mean that we're adding more SNPs to evaluate, to identify what's causing the significance. And so that's one thing we can do with the data that we have. Another, which is what I'm going to present today, is something that John Keel has been working on along with his reproductive data, and it's actually looking at copy number variation. Now, copy number variation, just like the name suggests, it means that you have variation in the genome for the copies of the copies that are within that genome. So for example, for a reference genome, we have segment A, B, C, and D, and that's what you would normally see in the animal sequence. However, when you have a copy number variation, you see alterations in that. For example, you can get a duplication. Here B is represented 2 times, or a triplication. You can also have a complex C and V where you have duplication, triplication, everything you can throw at it. You can also have an inversion of the genome. And then I'm going to present today is actually a deletion, where actually a part of that sequence is missing in the animal. So this is an example of what we saw when we were looking at pregnant animals versus open females. What the top graphs show is log-R ratio. And what that is you're basically looking at the intensity of the SNPs on the chip. And this is in chromosome 5. And so in a pregnant animal, we see normal intensity of the SNPs across this genomic region. However, in an open female, we see normal intensity, and then we get this drop. And then it goes back up to normal intensity, indicating that there is a deletion in that animal of this section of the genome. So we wanted to evaluate that a little bit further and find a way in which we can, without running the chip, which is fairly expensive, identify this deletion. So what we did is we utilized PCR. We basically know that the region that's deleted, we know the general size, what it's going to be, and so we generated primers that were outside the region and also within the region. And doing this, we're able to amplify different regions of the DNA. So in a normal animal, the entire section of that DNA is amplified, so the deletion is not present. However, this is an open animal. Outside the deletion is amplified, but when we look at the region within, no product is produced. So therefore, this animal, we conclude, has the deletion. So from this research, we were able to identify regions of the genome associated with reproductive efficiency, and for what I presented today, we focused on chromosome 5. We're able to replicate these results across multiple populations, and through further evaluation of these regions, we were able actually to identify a C and V on chromosome 5 in which there's deletion, in open females, at least across the 4 populations that we looked at. Now, what we want to basically get from this research is there's a lot of things that impact reproduction. Last year I talked about the Y chromosome. This year I'm talking about deletion on chromosome 5. And it'd be great if we could actually generate a panel that could be run across heifers before they are bred and be able to identify if they do have these mutations or deletions that affect reproduction, something that would be cost effective to the producer to run. So there's a number of individuals that helped with this research, from collecting the DNA, extracting the DNA, doing the analysis, and most of them are listed on this slide here. So with that, I'd entertain any questions you might have.